Hello and good day. My name is Derek Barmore Birowa, and today I will gonna report about my research on crop grains, specifically millet. So let's start. Introduction. Millets are a group of highly variable small-seeded grasses widely grown around the world as cereal crops or grains for fodder and human food. The Philippines has a rich biodiversity and many native plants like millets and species are consumed as staple foods. The Philippines mainly import millets with a total of 10,000 to 15,000 tons over the last 25 years. Botanical description. Millet is an annual plant of the grass family Graminae or Pucae with fine and long stems measuring from half a meter to nearly 2 meters high. 1,000 seeds weigh an average of 5 grams. Its fruit is a small grain seed with yellowish appearance, which is edible. Its light green leaves are long, wide and open, pointed, among which some lighter green spikes grow. They contain a couple of flowers. Millet can be best grown in spring season. Breeding and crop environment. Millet is becoming increasingly vulnerable to various climate-induced stresses, because of which the genetic and genomic resources will be important for improving the crop in the 21st century. Currently, sizable and untapped genetic resources exist that offer promise for millet improvement to biotic and abiotic stresses. The potential millet genomic resources such as molecular markers, genetic maps, and DNA sequence, but the data are scanty to support the efficient and accelerated delivery of the climate-smart millet varieties. Provided, a detailed analysis on the millet genetic and genomic resources aided interventions that could contribute to the 3 pillars of climate-smart agriculture, or CSA, for addressing millet production challenges under change in climate. It presented enriched information on additional useful sources of variation within millet genetic resources that have been screened for improving millet tolerance to various climate and induced stresses. It presents the novel opportunities for CSA that could come as a result of the recent availability of CGS data for revolutionizing the development of cutting-edge molecular breeding tools. Emphasis was placed on genome-wide based technologies such as genomic selection, or GS, gene pyramiding, and gene expression with the second-generation genomic biotechnologies, such as tilling and eco-tilling that are wanting and have received little attention. Cultural management and practices. Millets are one of the most farmer-friendly of crops. In fact, in many communities, millets are considered the lazy farmer's crop. One really needs to do just 2 things: go to the field to broadcast seeds, and then return after 3 months to harvest the grains. It requires almost no inputs, grow even in extremely low fertility soils, don't need deep plowing, can be sown with minimal tools or machines. Need only 1 weeding, if at all. With timely good rains at the right times, the farmer can expect to reap a good harvest. Using some improved techniques and practices, farmers can get a decent harvest even when the rains are not up to the par. Maximum millet cultivation happens in the Kharif period during the monsoon season. In areas that receive more than 10,000 millimeters of rains, many of the millets can be cultivated in the second season as a rabi crop during the post-monsoon early winter months. So millets are extremely resistant to pest attacks. This is a characteristic that comes in very handy when planning a mixed crop farm cultivated using non-pesticide management techniques. Post-harvest operations and processing. The main reason of less popularity of the millets food among the wheat and rice eaters are that millets have tough outer coat, related characteristics, flavor, and availability of processed products of millet similar to rice and wheat. For processing of cereals, many machines are available, but there is no well-verified method or process is available for making wild products from colored millets. Millet dehullers or other abrasive dehullers are used for decortication. Millets can be used as traditional as well as novel foods by cooking decorticated or whole and may be converted into flour using industrial or traditional methods, though it is required to find alternative uses possibilities. For significant utilization as food, inedible grains are converted into consumable form and also improving its quality by the processing. Millets can be processed into flour, porridges, popped, salted, ready-to-serve grains, sprouted cereals, roasted, and malt foods. Processing of millet grains initiate with the husk removal as it consists of hard seed coat. Post-harvest operations such as threshing, drying, cleaning, packaging, storage, processing, and transportation are as significant. This compromises the second half of activities following pre-harvest operations. Therefore, these are crucial processes in the whole food chain for the millet production. Issues and problems. The problem of environmental degradation is common for many crops, but particularly serious in millet. Population growth has forced farmers to shorten fallow periods, which in turn has resulted in declining soil fertility, and to expand millet cultivation into more marginal lands. Millet farmers are facing and the problems they encounter in terms of production. Both farmers consider climate change as the major challenge in terms of cultivation. They can no more follow the same planting season scheduled each year. The weather can be too dry or too wet. Both is not favorable for millet. In terms of production, the cost of labor are high. The farmers earn only 400 Philippine pesos for each unit of measurement of volumes, which is not enough to cover the production and transportation costs. They need subsidies and support from the local government. Recent advancements. Highly efficient and cost-effective transformation technologies are essential for studying gene function in millet. Millet can be transformed at very high efficiency by the methods are genotype dependent. Ideal transformation system needs to be extremely efficient, simple to perform, inexpensive, genotype independent, and give the required expression of the transgene. Thank you. And that would be all.